Good morning, all. The session is about the chromosomes. Chromosomes. The, the session is about the chromosome and the chromatin structure. So, in the previous session, we learned what the nucleolus and nucleus is. Let's see what a chromatin is. And this is the picture we have learned. And during the cell division, the chromosome undergoes maximum condensation during mitosis. In the cell cycle, during mitosis or during cell division, that is particularly in mitosis, the nucleus, the chromosome undergoes maximum condensation. And other times it will be in a less condensed form. So this is a replicated, you can see here, this is an unreplicated chromosome and this is a chromosome in the metaphase. And in the metaphase, the chromosome shows maximum condensation. Now, let's see what are the components or at which all phases of the cell division the chromosome condensation varies. So, the chromosome is made up of, so chromosome is the genetic material of a cell. Then the chromosome is made up of, or the chromosome is made of chromatin. The chromosome is made up of chromatin, and what the chromatin is composed of DNA and histones. Chromatin is composed of DNA and histones, and main proteins which are found associated with the chromosome are called the histones. And the histones are the proteins which help in the intact packing of DNA inside the nucleus. So chromosomes are found inside the nucleus. Chromosome is made up of chromatin. Chromatin is equal to DNA plus proteins. And the main proteins which are found in the chromatin are the histones. Now, the chromatin remains highly extended or highly loose. The chromatin will be in a loose or an extended condition in the undividing cell. But after division or during the interphase or during the mitosis, the chromatin gets maximum condensation and they remain in a condensed or compact condition. So, based on this, there are two basic types of chromatin, that is euchromatin and heterochromatin. Chromatin exists in two forms during the cell cycle. They are euchromatin and heterochromatin. Let's see one by one. First is euchromatin. Euchromatin, in euchromatic condition, the cells or euchromatic condition, the chromatic fibers are in a loose condition, or it will not be tightly packed. Or euchromatic condition, the chromatin is less coiled. So in this picture you can see the green color structures are the chromatin and the orange color structures are the proteins or the histones. So this is a chromatin. And these orange structures are proteins. So during cell division, or the chromatin fibers will be in a loose condition inside the cell. And this euchromatin is euchromatic condition, the transcription of the proteins takes place inside the cell. So euchromatin is the chromatin in which the chromatin is in a highly dispersed or in a loose state during the interphase. And in this phase, the chromatin is active for transcription. So euchromatin is the active phase. That is the main difference. Euchromatin is the active chromatin or active phase where the protein synthesis or the RNase protein synthesis and RNA synthesis happen in a chromatin when it is in a euchromatic condition. That's what's known as euchromatin. Eu means true chromatin that is involved in RNA synthesis and proteins. That's called euchromatin. So chromatin exists in two forms inside the cell during cell division. One euchromatin, two heterochromatin. Euchromatin means true chromatin which is involved in the transcription of proteins. They are active proteins and they are found in a loose condition. You can see here the chromatin is free here. It's not bound around the proteins. The chromatin is free. See this green colored part, this chromatin is free for transcription. That's what's known as euchromatin. Now, heterochromatin. See the difference? In heterochromatic condition, it is tightly packed. The chromatin is so close to each other and there is no loose chromatin, or the chromatin is not exposed for transcription. And in heterochromatic condition, the chromatin remains in a highly condensed state. It's not loose. It is in a highly condensed state. The chromatin is in a highly condensed state. That is, during interphase, the chromatin or throughout the cell cycle, the chromosome will be in a highly condensed state. That is what is known as heterochromatin. So, the chromatin is not at all free for transcription or translation. And the heterochromatic condition is of two types. Heterochromatic condition is of two types. One is constitutive heterochromatin. One is constitutive heterochromatin and two facultative heterochromatin. Constitutive and facultative heterochromatin. So, in constitutive means continuous. Constitutive heterochromatin means continuous heterochromatic condition. That is, the chromosome or the chromatin will remain in a highly condensed state in all time of its life. Always. Constitutive means always, or continuous. Constitutive means always or continuous. The chromosome will be in a heterochromatic or inactive or condensed state. Constitutive means continuous heterochromatic condition, or always it is in a silent condition. That's what is known as constitutive heterochromatin. And the examples of constitutive heterochromatin are, we learned in the previous class, that is centromere. The chromatin found at the centromere and telomere are constitutive. So, the chromatin which is found at the centromeric region and that is which is found at the tip of the chromosome are always in a heterochromatic condition. So, heterochromatic condition remains in a highly condensed state in all cells at all times. That means continuous silence. Where it is found at the centromere and in the telomeric region. And so these chromatin or this DNA is in a permanent silent condition. Then they are situated in centromere and telomere. Then telomere and this region where there is constitutive heterochromatin will have continuous repetitive sequence. Hope you remember we learned about telomere last class and we said, can you tell me the answer? Which are the continuously repeated sequences at telomere? You can put the answers. The highly repetitive sequences, which was the highly repetitive sequences at the telomeric region. So, the constitutive heterochromatin have highly repeated DNA base pairs or highly repeated sequences. And we learned in the previous class about the telomere where it is having a continuous highly repeated sequences. They have repeated sequences and this region does not have a chance of protein coding genes. They don't code for any proteins, but they are in a highly intact form. And actually, because they are present in telomere, they may be preventing the wear and tear of the chromosome during cell division. And when the genes are active in the normal stage, when moved to this position, and another phenomena of this constitutive heterochromatin is, because nobody have given the answer for the repetitive DNA sequences or at the telomeric region, I didn't get any answers. The repeated DNA sequence at telomere. Which are the repeated DNA sequence at the telomere? Nobody has answered so far, so I will put the answer. That is the repeated DNA sequences at the telomere was TTAGGG. The repeated DNA sequence at the telomere was TTAGGG. TTAGGG. So let me tell once more about, yes, another phenomena of this heterochromatic DNA is that when heterochromatin is inactive stage, permanent silence, that is heterochromatic region. The peculiarity is inactive, 1 inactive, 2 condensed, 3 permanent silence, then they are devoid of protein coding genes. And another peculiarity of this constitutive heterochromatic region is that when a active chromosome, or when a gene which is active, when it comes and sits near this heterochromatin, when an active gene moves near to this heterochromatin, what happens is that the active chromatin will become inactive, and that is known as position effect. Important, position effect. Position effect. Position effect means position effect. When an active gene moves near to this heterochromatin, when an active gene in a normal state move near to this heterochromatin, that gene will become inactive, or that gene will become silent. That is known as position effect. Important. Position effect is when an active gene moves towards a heterochromatic gene, it will become inactive. That's known as position effect. So, constitutive heterochromatin is a highly condensed state. Constitutive heterochromatin, the different characters are, let me brief once more, one by one. 1, highly condensed state. Second point, permanent silence. Third point, they are located in regions like centromere and telomere. Fourth point, they have repeated sequences, like in telomere we learned TTAGGG. They have highly repeated sequences. Then, 4, they are devoid of protein coding genes. These heterochromatin will not code for any proteins, or constitutive heterochromatin will not code for any proteins. And the 6th position is the important, that is known as position effect. When an active gene moves near to this heterochromatin, it will become inactive. That's known as position effect. So these are about the constitutive heterochromatin. Next, about the facultative. Facultative, the name itself is clear for you. Facultative. Facultative means occasionally only they will become inactive. Facultative heterochromatin means they are inactivated only during certain phases. Facultative means occasional heterochromatic condition. Facultative means during the cell cycle, the chromosome or chromatin will become inactive only during certain phases. That's known as facultative heterochromatin. Then, the transcriptional activity of this facultative heterochromatin is in the function of time, that is depending on the need of cell. For example, If a cell needs a protein at a particular time, at that time only the chromatin will be active. At all other times it will be in a silent condition. So that is known as facultative. At times only the chromatin will become active. That's known as facultative heterochromatism. Now, the example of facultative heterochromatin are, one important example is X chromosome. So, in females, we have two X chromosomes. And among this X chromosome, one X chromosome is inactive. But only one is active. And this is known as facultative heterochromatism. Heterochromatin. Depending on the need of the cell, the X chromosome will be active, and this inactivation of one X chromosome in females is actually for the dosage compensation, because in humans males have one X and one Y, but females have two X chromosomes. So if both the X chromosomes are functional, what happens? There will be more number of X proteins. The proteins which are produced on the chromosome X will be more in females. In order to maintain the balance between the X proteins, the proteins from X chromosome between the male and the female, one X chromosome is inactivated in case of adult females. But during what happens with this? And this condensed X chromosome in case of females, it is called bar body. This heterochromatinized X chromosome in females is called bar body. The heterochromatinized X chromosome in females is called bar body. And the bar body is the condensed X chromosome in females. So, there is a balance between the proteins of X between male and female, and you can see a bar body in any of the cells. In all cells, the cheek cells, in all cells of females there will be a bar body, but males will not have a bar body. Why? Because they have only one functional X chromosome. So males don't have bar body, only females have bar body. In females, the bar body significance is the inactivated X chromosome to maintain the balance of X proteins from X. Now, because this was discovered by the inactivation of X chromosome and heterochromatinized bar was studied by Mary Lyon, this is also called lionization. This is also called lionization. This is also called lionization. Lionization is the inactivation of one X chromosome in females or mammals, or in females. That is X inactivation. And X inactivation is in embryo is in a random form. The random manner of X inactivation takes place in the embryo to maintain the balance. That is all about the heterochromatin. So heterochromatin means the condensation of chromosome. Heterochromatin means only less part of the gene is active for transcription. The other chromatin will remain condensed during the interphase. That is known as the condensation of chromatin in the interphase of cell cycle is called heterochromatin, and heterochromatin is of two types, constitutive and facultative. Constitutive means continuous silence. They are found in telomeres and centromeres. Facultative means occasional silence only, and that is example is X chromosome in females. Now let's see how the chromosomes are arranged inside a cell. So chromosomes, we learned that the chromosome of the eukaryotes. The peculiarity, what is the peculiarity? The presence of nucleus is in the case of only eukaryotes. Eukaryotes only have a true nucleus. Eukaryotes only have a true nucleus. So the chromosomes of eukaryotes is made up of chromatins. chromatins and associated proteins. So chromatin is equal to, we learned that is DNA plus protein. Chromatin is equal to, chromosome is made up of chromatin, chromosome is made up of chromatin, and chromatin is equal to DNA plus proteins. Chromatin is DNA plus proteins. Chromatin is DNA plus proteins. So, how this is found associated? And the proteins which are found inside in the eukaryotes are called histones. The proteins in the eukaryotes are called histones, and that histones is a peculiarity of eukaryotic cell. The histones are unique proteins which are found in eukaryotes. Why? Because, can you tell me why histones? I said histones are the proteins associated with the DNA of eukaryotes. But histones are found only in eukaryotes. It is absent in prokaryotes. Can you tell me what's the reason for that? So the histones are unique proteins which are present in eukaryotes. And why? Because the nuclear membrane is present, or nucleus is present only in eukaryotes. Nucleus is present only in eukaryotes, or nuclear membrane is present only in eukaryotes, but prokaryotes are devoid of this nuclear membrane. So, histones of eukaryotes are of different types, are of 5 different types, that is H1, H2A, H2B, H3 and H4. And these are the abundant proteins which are associated with the eukaryotic cells. And the number or the amount or the mass of histones is equal to the amount of DNA. So, then the 5 different types of histones are H1, H2A, H2B, H3 and H4. Let's see, this is actually the structure of histone proteins. This is DNA which is wound around the histones. These are histones. The gray color structures are histones. This is H1 histone, and inside this DNA is found coiled around the histones inside the nucleus. So actually the histone hold together the DNA inside the cell. Then the peculiarity of histones are: 1, it is unique to eukaryotes, and second is they have high amount of the basic amino acids like arginine and lysine. Histone ഒരു പ്രത്യേകതയാണ്. ഹിസ്റ്റോണിലെ ഏറ്റവും കൂടുതലുള്ള amino acids എന്ന് പറയുന്നത് arginine lysine. That is basic amino acids. And why basic amino acids? These basic amino acids, the presence of more basic amino acid help in binding to the DNA molecule. ഈ ഹിസ്റ്റോൺസിനകത്ത് basic amino acids ആയ arginine, basic amino acids ആയ arginine lysine ആണ് കൂടുതൽ കാണുന്നത്. Why? This basic amino acid help in binding strongly to the negatively charged DNA. The DNA is negatively charged. So, the basic amino acids in the histones help in the close interaction of histones and the DNA. So, histones are unique proteins which are found in the eukaryotes. What is the function? The DNA is wound around the histone proteins. The histone proteins are made up of mainly basic amino acids. Which are the basic amino acids? The basic amino acids are arginine and lysine. Then why? The DNA is acidic DNA molecule. That's why we say nucleic acids. Deoxyribonucleic acid. That means it is an acid. and that facilitates, so this basic amino acids in the histone help in the binding of the histones to the DNA. So there are 5 main types of histone proteins: H1, H2, H3 and H4. Then the total mass of the histones, you can see here, the H1 histone is found outside the core. This is known as the core. This region is the core region, and inside the core region the 8 histone molecules are found, and one, that is the H1 histone, is found outside this core region of the nucleosome. So the H2, H2A, H2B, H3 and H4, they form an octamer. That is, they are found in 2 numbers and they are form an octamer. But the H1 is found outside this core region. H1 is found outside the core region and it is associated with the linker DNA. So that's all about histones. Histones are unique proteins of eukaryotes. There are 5 histones: H1, H2A, H2B, H3 and H4. The 4 H1 is found outside the core. All other are found in 2 in number and they form an octamer. Now let's see why. What is the importance of this histone, or what is the importance of chromosome packing? So chromatin, I said, it is made up of chromosome and DNA. And the very important thing is the nucleus is very small structure. The nucleus is very small and it has a diameter of 5 to 10 μm. The diameter of nucleus is 5 to 10 μm only. The diameter of nucleus is 5 to 10 μm only. That it varies according to the cell. That is why read this written. 5 to 10 μm is the diameter of a nucleus. But it can hold the DNA which is of 2 meter in length. The DNA, if you stretch the DNA, it is very long. That is, it comes to about 2 meter approximately. And this 2 meter structure has to be, just imagine, this is only μm, micrometer, 10 raised to minus 6. So this 2 meter long DNA has to be kept very intact in a cell. That is the importance. That is what is known as the chromatin organization, or different levels of DNA packing. That is the significance of DNA packing. So the 2 meter long DNA has to be packed inside a nucleus which is having only 5 to 10 μm diameter. That has to be done. That is the that is a challenge. What we say as nowadays, that is a challenge of this this chromosome. So it has to be folded in very tightly so that it can be kept inside the nucleus. 2 meter long chromatin has to be kept inside a nucleus which is very, very small in diameter. Now let's see one by one the different levels of chromatin organization. First one is called the nucleosome. Nucleosome. And this histone proteins, we learned the histone proteins are 5 in number, and this histone proteins help in keeping the DNA intact inside the nucleus. Let's see how. One reason we told they are positively charged or basic and the DNA is negatively charged. This is known as a nucleosome. Let's see, the DNA wound around the octamer is called a nucleosome. And this nucleosome structure was first described by Roger Comberg. Roger Comberg first described the structure of this nucleosome. Nucleosome. This is nucleosome. Nucleosome means the DNA plus the core or the octamer of histones is from the nucleosome. And nucleosome model was first used to explain the structure of the DNA arrangement. Just see, this is the first order packing of nucleus. Here you can see the DNA, and DNA is wound around the nucleus like beads on a string. Like beads on a string, the nucleus, the DNA is wound around the octamer, and this stage is called the nucleosome. That is the first order of packing of DNA inside the nucleosome. So nucleosome is the basic structural unit of a chromosome. Nucleosome is the basic structural unit of a chromosome in which the DNA is wound around the octamer, and the nucleosome consists of a nucleosome core particle, and it is formed of histone octamer, which is wrapped around the DNA helix, and this DNA helix have a length of about 146 base pairs. 146 base pairs of DNA is wound around a histone octamer, and the histone octamer consists of 8 histone proteins. The nucleosome core particle, here you can see this is known as the linker DNA. In between nucleosome, this is one nucleosome, another nucleosome, and another nucleosome. In between there is a strand of DNA that is called the linker DNA. That is the linker DNA, and this is known as first order of packing of nucleosome. This is the first order of packing of nucleosome, and the linker DNA have the linker DNA will be having 60 base pairs. Linker DNA will have 60 base pairs, and the DNA which is wound around this octamer will be having 146 base pairs. So together the DNA of a core particle will form about 200 base pairs. Hope it's clear. So nucleosome is the first order of packing of DNA inside the cell. How nucleosome means DNA wound around a octamer. Which are the proteins helping this winding? They are the histones. Histones are 5 in number. So here you can see the histone, they have octamer. Histone octamer is found here, and one histone protein is found outside this core, that is known as the link, that is H1. H1 histone is found outside. They have no role in this octamer region. So DNA is wound around the octamer, and the length of DNA which is wound around the octamer is 146 base pairs. Now, in between two nucleosomes, you can see a linker DNA. That means it links two octamers. And this linker DNA, how many base pairs are there? 60 base pairs. And in total, this two, that is 146 base pairs plus 146 base pairs plus 60 base pairs form the 200 base pairs. That means from this region to this much region, it is 200 base pairs. Hope it's clear. That is 200 base pairs. This is the first order of packing of nucleosomes. First order means it looks like beads on a string. So first order packing of chromatin is called nucleosome. Nucleosome means histones plus histone octamer plus the DNA, and the coiled DNA which is locked in place of H1 protein. What is the purpose of this H1 protein? Actually, I said one H1 protein is found outside the octamer. See, if this is a bead, this is a DNA, this is a DNA, you have a solenoid-like structure, and histone is found here. H1 is found outside this nucleosome. So this is a DNA and this is the octamer, and one H1 is found outside. And actually the function of one H1 is, this H1 will prevent the coiled DNA is actually locked. this DNA is locked by this H1 protein. That is the significance of H1. Hope it's clear. One histone is found outside the core. The function of H1 is to prevent the uncoiling of the DNA around the octamer. That is H1. This is known as the nucleosome, and the term chromosome is also applied to this complex structure. So chromosome, this is also called a chromosome or a nucleosome. So nucleosome is the basic or the first order. How nucleosome, how a DNA is packed inside a long DNA is packed inside a small nucleus. It is wound around the, like simply like we wind the thread around a block or something. Likewise, it is wound around the histone proteins. Histones are unique to eukaryotes, and out of the histones, one, that is H1, is found outside. Why? It helps in locking, or it helps the unwinding or uncoiling of this DNA. That is the first order packing. Then, many number of this nucleosome join together to form a structure called polynucleosome. So polynucleosome is, this is called a polynucleosome. Polynucleosome means many nucleosomes. A group of nucleosomes is called polynucleosome, and it helps in, this polynucleosomes look like a beads on a string. Now, next level or second order packing of DNA, and this actually the first order will only reduce the reduce to very less number, or less condensation happens as a result of this nucleosome. Again, it has to be condensed. See, this has come to only this levels, and again it has to be condensed. And next level are the higher levels of nucleosome or chromatin organization. Nucleosome is the lowest level or the first order of chromatin organization. That's over. Next, we'll see the higher levels of chromatin organization. How more condensation is happening. So, the 11 nanometer beaded structure, it undergoes much more folding. That is known as second order. In second order, what happens? The DNA is bound, or the nucleosome again undergoes twisting. That is known as a 7-fold reduction takes place to happen to this chromatin. And this nucleosome structure or the polynucleosome will again take a twist and turn. They will again take a twist and turn to take more condensation. And let's see how it is happening. This happens, that is, this 30 nanometer fiber appears to form a condensation of 11 nanometer fiber, and that is known as solenoid structure. This is called a solenoid arrangement, and in this solenoid structure, what happens? This is the solenoid structure, means many nucleosome again undergo twist and turns, or scaffold reduction. That is, they undergo twist and turn to form a solenoid structure. And this solenoid structure, or in one solenoid, 6 to 7 chromatosomes are present. 6 to 7 chromatosome. Chromatosome means what? This group, this nucleosome, this polynucleosome is called a chromatosome. So 6 to 7 chromatosomes, they undergo twist and turn to form the solenoid structure. And the maintenance of this filament depends on the interaction between the H1 histone molecules. Actually, this H1 histone molecules have an important role in holding this nucleosomes together. H1 protein always, H1 histone help in the compact packing. They will hold together all the nucleosomes. And with the attainment of the solenoid structure, the DNA will achieve a 100-fold condensation. A 100-fold condensation, that means 100 times. The condensation takes place as a result of the solenoid formation. The DNA achieves a 100-fold reduction, means tightly packed. Once it is DNA is wound around the nucleosome, again it undergoes twist and turn to form a more compact structure. That is known as 100-time reduction of the length happens as a result of this solenoid formation. And in the next stage of hierarchy, in the third stage, it is the scaffold or the looping. In the next stage, the 30 nanometer protein, and here you can see this is a 30 nanometer chromatin fiber, and this 30 nanometer chromatin fiber will undergo a series of coiled loops. So this solenoid will form a coiled loop. So see this part, this much part only constitute for this much region of the solenoid. See the structure, this much DNA is tightly packed in this much part, small part. Again the reduction of the condensation takes place. See this image. So this much part means, or DNA means, or chromatin means this much only little amount of this solenoid. So in the coiled loop, as a result of the coiled loop formation, the packaging of DNA molecule take place, and each DNA molecule, each loop contains 10 to 100 kilobases of DNA, and that is linked to protein scaffolding. This is known as protein scaffolding. Means simply looping. A simple looping is formed, and as a result of this looping, each loop will contain 10 to 150 kilobase pairs of DNA. KBP, kilo base pairs of DNA, and this is known as protein scaffolding. So it again reduces the area which is occupied by the long 2 meter DNA. So what happened? First the DNA was wound around the nucleus, eptamer, then that eptamer or that polynucleosome again undergo twist and turn, and they took a structure called solenoid-like structure. The solenoid underwent what? A coiling or a scaffolding to form a scaffold loop. And the proteins, there are a network of non-histone proteins associated with this scaffold formation. During scaffold formation, when a DNA has to undergo scaffolding or folding, many proteins are involved, and there are the non-histone proteins to which the 30 nanometer fibers are attached as loops. To certain proteins, the 30 nanometer fibers get attached, and these are the non-histone proteins or the scaffold proteins. Scaffold proteins help in the binding. And in interphase chromosome, the 30 nanometer chromatin loop from the extended protein scaffold is found, and this they will undergo folding and scaffolding like this, and they are packed inside the metaphase chromosome. Only this much part, this much long is the chromosome, but only this much part forms this scaffold. Again, lot many scaffolding happens, and they will result in the formation of this metaphase chromosome. So in metaphase chromosome, the scaffold is folded into a helix and form a highly condensed structure, and the formation of the loop help in the condensation of chromosome, that is the loop configuration is almost 300 nanometer thickness. So further the folding and the twist and turn of this happens, and thus it is kept inside a metaphase chromosome. This is about the packaging of chromosomes. So, with the help of this histone proteins and the non-histone proteins, the long chromosome is packed inside a small nucleus which is having a less or a small diameter. Which are the different levels? The basic level is nucleosome. The second level is the second level is actually the solenoid phase. Second is solenoid. Then third is the scaffolding or the folding of this solenoid structure, and the scaffolding will end up in the formation of a metaphase chromosome. And this is how the chromosome is tightly packed or condensed inside the chromosome or the chromatid. So with this I conclude today's session, and this session was about the chromosome organization or chromatin organization and different levels of chromatin condensation inside a cell. Hope it's clear. Thank you.